protein expression levels and the selection pressures on those in primates. Thanks.
So thank you very much for coming to my talk. I want to thank the organizers for giving me the opportunity to present this work. So I thought I'd start this talk off with a broad motivating question. It's the question of what makes us human. So there are extensive phenotypic differences between us and our closest relatives. And an important goal of evolutionary genetics is to understand what genetic mechanisms underlie these differences. So there are two broad contributors to phenotypic differences. There are mutations in DNA sequence that lead to differences in gene function, which in turn lead to a difference in phenotype. And then there are differences in gene regulation that eventually, mutations in DNA sequence that lead to differences in gene regulation, which in turn lead to a difference in phenotype. And in the evolution of human, it's been thought for almost 40 years now that mutations that lead to differences in gene regulation likely played a more important role. This is primarily due to the scarcity of amino acid sequence differences between us and our closest relatives. So to understand the mechanisms that underlie phenotypic differences across species, you have a mutation in DNA that leads to a change in gene regulation and a difference in phenotype. One of the first steps is to identify genes that might be differentially regulated. And here, technologies like gene expression microarrays and more recently high-throughput sequencing have truly revolutionized the ability to do this. So prior studies have largely focused on transcriptional regulation. So using technologies like ChIP-seq, methyl-seq, RNA-seq, using high-throughput sequencing. And they've identified many genes and regulatory processes that have transcriptional differences. Yet two questions stand out. Do these actually contribute to phenotypic differences? There's a long way to phenotype. And how do these actually achieve this? So proteins offer one step closer to phenotype. So you have to have a mutation in DNA that leads to a change in transcriptional regulation, which must show up at the proteome to be functional, which in turn contributes to some sort of phenotypic difference. So proteins are independently regulated by a number of known mechanisms involving post-translational modification, which could alter their degradation rates, excretion, and so on. And here, you can't high-throughput sequence your proteins. It'd be wonderful if you could. The technology changes to high-resolution mass spec. So the goal of this study was to really ask this question: how does protein abundance differ between humans and chimpanzees? And how do they relate to differences in mRNA expression levels? And can we use these data using rhesus as an outgroup to find genes and regulatory processes that are of adaptive significance to human? So we're not the first to look at this. So if there was two studies which looked at rough estimates of absolute protein levels across diverse taxa, and they found that the correlation between these levels across these taxa tend to be more similar. And the mRNA, the absolute estimates of mRNA levels are less similar by linear correlation. So this work has a couple limitations. These absolute estimates aren't accurate enough. They're using spectral counting. They're often not accurate enough to identify single genes that are significantly different. Many different array platforms were used, which is really quite worrisome. And it's not entirely clear what this means for closely related species like primates. So we conducted a study in which we used lymphoblastoid cell lines derived from 5 individuals from each species, from human, chimp, and rhesus. And we analyzed the data in the context of orthologous gene models across these species. So I might use a term divergence in this talk, and that term just refers to the log2 ratio between human and chimpanzee. And this ratio is measured for mRNA levels as well as protein abundance. So if I say divergence, it's this log2 ratio. So we quantified protein levels using a technique called SILAC, in which we grew up an independent cell line and labeling media as the internal standard, right there. And we spiked that into each of the 15 samples that we analyzed. And we used that as a common reference to derive protein levels across individuals for each gene, so relative quantification. So the protein data obviously was the limiting in terms of coverage of the number of genes. So on the left is a bar plot requiring quantifications in more and more individuals in each species. And in purple are the genes we analyzed. These are generally medium to highly expressed. And if you relax the species cutoff, you get the genes in green. And then if you merge all 15 data sets, you see that we can get a peptide that goes into the much lower expression range. So the first thing we did with this data was conducted a differential expression analysis in which we asked which genes are differentially expressed at the RNA level as well as the protein level. So when we did this, we found at FDR 1%, many more genes differentially expressed as mRNA than as protein. And on the left are the full changes for each of those categories. And this can be due to a number of possibilities, each of increasing levels of interest. So it could be because the protein data are just really noisy. And we looked into this. They're noisy and inaccurate. So we chose SILAC for a reason. So SILAC, you get, if you titrate in a peptide with a known femtomolar amount, you get a linear response that actually matches your expected value. So this is in contrast to these techniques called ITRAC and TMT, which use isobaric labeling. They're in the best case, they cause ratio compression because of the physics of the mass spec. And then in the worst case, they cause ratio distortions. You get quantifications that are completely off. So the next thing is the noise in the measurements. So what we did to assess this was we looked at essentially RNA, this divergence measured from distinct sets of exons. So this is a median value from two sets of exons from the same gene. So one point is a gene here. And we asked how consistent are they? And we find that if you take peptides and map them back to distinct exons, you find they're generally, the quantifications are more consistent. So it's not that the protein data is extremely noisy. In fact, it could potentially be actually better quantitatively. So this leaves two more possibilities. It's just that it could be that just protein levels are more variable between species. And one way to assess this is to look at the interquartile range of the divergence measure, measured as mRNA and as protein. And you find that this interquartile range is no larger at protein than mRNA. Another thing you can do is essentially, so I use the replication in our study to measure this divergence ratio for mRNA and then for protein. So now you get the variability for protein and then for mRNA. You can also write this is sort of a more convoluted way of writing this, but you can also write this as in a linear model where you test for a species by technology interaction. So now we're not looking at these genes in these different categories. We're looking for genes that are significantly off the diagonal, those where this divergence measure is significantly different across technologies. And when you do this, you find many more genes where mRNA divergence is higher than protein divergence, than there are genes where protein divergence is higher than mRNA. So you can see we have error bars here on no divergence. And here we have error bars on the divergence measured as mRNA. So ultimately, what we're getting at is we're measuring gene product levels using two different technologies. And these two approaches have their own sources of noise and biases. So we wanted to confirm that this sort of observation here was robust to a number of quality controls. So we applied various restrictions on the number of individuals the protein was quantified, cutoffs on this interquartile range, peptides quantified per individual, RPKM cutoffs, renormalization, removing highly expressed mRNAs, and we still see this pattern. We also transformed our data into spectral counts using the signal from the light unlabeled cell line. And we see what these other studies have actually seen before, which is along this absolute range, mRNA levels are less correlated protein levels. So the effect is much less pronounced because we're looking at two very similar species. So using these lines of evidence, we argue that over evolutionary time, protein levels are under greater evolutionary constraint than our mRNA levels. We have several lines of evidence to support this. So this is broad analysis of a broad set of genes. What if we look at the individual genes that we have quantified? So in part 2 of the study, we analyzed individual genes, and the goal of this part of the study was to find genes and regulatory processes that were of adaptive significance to human. And the approach we took was to identify molecular signatures of selection. So the sort of standard approach for doing this is to look for patterns of nucleotide diversity. And if you can find these patterns across individuals in a species, you can identify regulatory regions as well as genes that might be of adaptive significance to human. Yet another approach is to look at gene product levels across species, and you can use this to identify candidate genes as well as regulatory processes and pathways. So there are two broad approaches for looking for signatures of selection of on gene regulation. The first set of approaches assume a null model and test for deviations from this null. So there's a body of literature on building these models. Yet it's impossible or impractical to measure the necessary parameters of these models, like mutation accumulation rate in non-model systems. The second approach is to essentially look at these cross-species expression pattern and rank them and how consistent they are with a particular mode of selection. This is, of course, heuristic by comparison, and it provides a candidate set of genes for further study. So we looked at two types of selection, stabilizing selection, in which you have selection against both extremes. Here the trait would be protein level, or directional lineage-specific selection when you have selection against one side of the distribution, across only in one lineage. So we essentially identified patterns like this to identify genes that might be subject to stabilizing selection. So you have low variation across species, as well as low variation within the species. And you can rank genes based on their intra and inter-individual variation. And genes at the top of this list are very likely candidates for stabilizing selection. So when we do this for all genes using mRNA, and we do this for protein in blue, we find that, as other people have observed, the mRNA genes, the genes that come up using mRNA, are correlated with these measures of regulatory constraint, like non-synonymous, dissynonymous substitution, number of protein interactions. Yet when you go to protein, the effect is always more pronounced. So it's also the case for these measures as well as several others, but for some reason they're not coming up here. But nevertheless, we see this for several other measures like UTR length and expression in number of tissues. So this would make sense because selection is not acting directly on mRNA levels. It's very likely acting on protein, which are more directly coupled to phenotype. So we also look for these patterns where you see a change within a lineage. So here, this gene DDX24 is the same to the outgroup rhesus, and it's increased in the human lineage. And here on the right, human and rhesus are the same, and there's a chimp-specific decrease in this gene. And among genes where we found a human-specific change, the genes are involved in transport and localization, which are truly post-transcriptional, post-translational processes, rather. And you can't see this using mRNA. So now we're sampling two regulatory layers, transcriptional regulation as well as protein regulation. And now we can identify genes with more complex, potentially under more complex evolutionary scenarios. So genes where there's a post-transcriptional or post-translational change. So here's an example of a gene where we see a human-specific change at the protein, but not at the mRNA level. So at the top here is the average read coverage for each of the species, and on the right are the per individual measurements we get. And on the bottom, I've mapped the peptides back onto the gene model, and these are the relative quantifications. So you can see clearly chimp and rhesus are the same, and there's a human-specific increase across this gene. And such a pattern would be consistent with a human-specific change in post-transcriptional or post-translational regulation. Yet we see many genes where there are no difference at the protein, yet a large difference at mRNA. And such patterns could arise out of two potential hypotheses. First, the proteome or the protein-protein interaction network acts to buffer a lot of these changes. Or perhaps more interestingly, compensatory evolution, where you have a change that caused the difference in mRNA, which made humans less fit, and a second compensatory change that acts to bring protein levels back. So we find genes in our data consistent with this pattern. So here is the RNA-seq read coverage with human-specific decrease, and chimp and rhesus are the same. These are the per individual measurements for mRNA. And then across this gene, if you of the many peptides quantified, you see absolutely no change across all three species. And these are the per individual measurements. So just a few broad observations. You have mutations in DNA that lead to changes in mRNA regulation. It appears many of these differences in mRNA appear to be neutral. They don't show up in the proteome. Perhaps it's speculation at this point, but I like to think of this as potential cryptic variation in the proteome. Perhaps at one time in the future, it'll become of adaptive significance to human. Yet it's not sufficient to just look at transcriptional regulation, so just RNA-seq or related technologies. You have to really look at proteins to fully understand these processes. So we see differences in post-transcriptional or post-translational regulation. And we are working on collecting the point in between using ribosome profiling, so translational change. So we can really nail it down to protein regulation, so all of these combined, there's still many more steps to leading to difference in phenotype, but these studies are productive in identifying individual genes and regulatory processes. So with that, I'd like to acknowledge my postdoctorate mentors who had the very deep pockets to collect this data. Michael Ford at this a CRO company who collected all our high-res mass spec data. All of it's available here for download. Darren helped with RNA-seq, Amy grew the cells. And I've started my own lab. I've been, CBC at Maryland has been generous to host me. And I'm interested in joint quantitative transcriptomics, proteomics data sets. I mean, really, it's based on this idea that I strongly believe, which is to fully understand how genes are regulated. It's not sufficient to look at transcriptional regulation. You have to really look at them as translated proteins as well. And here's the link for the data. And I'd love for you guys to fully explore it. It's a full data set. You can analyze the spectra from the ground up, as well as download processed tables. And with that, I'd like to thank you for listening.
Hi, it's very nice work. One thing that you didn't discuss is relative rates of degradation. So the amount of either the RNA or protein that you quantify will be a function of the rate at which it's synthesized and the rate it degrades, and proteins certainly vary substantially in their rate of degradation. So an alternative that you didn't mention is that you require a certain amount of protein that is what's giving rise to the molecular phenotype. Some of those are just inherently unstable, and so the cell has to ramp up the amount of transcription translation to maintain the protein level the same, and that's why you see so much variation in the RNA. Do you think that's reasonable? Is it something you've thought about?
It's actually really hard right now to get degradation rate measurements. You have to use a pulsed SILAC type experiment, which is even more expensive than steady state levels. So having those measurements in human would be just incredible. A time course where we can get degradation rates as well as synthesis rates to really nail down where in the regulation of these genes the primary contributors are. So I have thought about it, but it's the experiments are at this point expensive.
Is it possible to do it just in a couple of your outliers so you can see, the extremes of your distribution?
We could, when we didn't in this study, but that would be a wonderful follow-up work. Thanks.
Hi, very nice talk. I'm wondering, to make it even more complicated, you're monitoring messenger RNA and you're monitoring proteins, but what about alternative splicing and isoforms? How much does that add to the complexity? Does it increase your error bars, or is it an opportunity?
It's an opportunity that I hope my group will explore. So majority of the splicing signal is across tissue types. Here we used one single model system, which is a single cell type lymphoblastoid cell line. At present, there's no good quantitative method for identifying isoform switching and proteomics data. We are working on a couple candidate techniques, so that would be really cool. And I think we can get the coverage high enough too. So it would be really cool.
Hi, so I have a question about the hypothesis that it's the compensatory evolution hypothesis, the idea that like gene A stopped or made less of the protein, and so gene B came, increased to compensate. And the question is, if it happens in that order, how does the lineage sort of survive in between? And is it possible that instead of doing it that way, it was sort of, more like gene B made its change first, and so, you have both, and allowing A to sort of either drift out or get whatever advantage you got from removing A or lowering A?
So I think testing this compensatory hypothesis I think yeast would be a better system to do it in, where you can evolve yeast strains or bacterial strains. I think our work would say that if some of those results were found in yeast or in bacteria, that they're important to human as well. So I don't think we just point out potential hypotheses for these patterns. We don't know if they're true. So they're candidate genes for study. So this is all we can do with the systems biology at this point. So answer is I don't know.
Thanks.
Isaiah, very interesting. What were the cell lines grown in? What media? Were they grown in a rich media?
So it's a model system. So these are EBV transformed cell lines. They're grown in DMEM, they're transformed EBV,
Okay.
EBV, and they have all the caveats of the NEBV system, so the HapMap cells.
That's fine. You measured though, and you said, well, there's a good deal that may be cryptic in terms of the protein levels and what they might be doing, right? So this could all just be reflecting the fact you're growing these things in rich media. Right, and not at all in the system that, at least I'm not bathed in.
Yeah. So
Yeah.
I agree.
Towards us though, right? Do you think that the next approach should really be things like pulse, SILAC, or just explore different conditions?
So there are two ways to take this work. Obviously, more regulatory profiling, or looking at actual tissues. And tissues are a little tough in primates because of the ethical issues of obtaining them. So I think if we could explore those couple of dimensions, I think it would really build on what we've built here.
And do you think that's the future? Is through, to go through tissues and explore it that way?
I think both would be really
Awesome.
really promising. I think. Thank you.